This is the lesson on the biochemistry of lipids. These are the objectives. We're going to look at the functions of lipids, first of all, followed by a recap of your GCSE knowledge, hopefully, and how that links into the formation of tags, and I'll talk about those in a minute. We'll then look at the condensation reactions that happen to form the types of lipid that we're going to talk about. And obviously the reverse of that is hydrolysis, which we've come across twice now. We're going to look at how different lipids can be saturated, what we mean by saturated, what we mean by unsaturated. We're going to look at the biochemical test for lipids, the structures of phospholipids and membranes, and then a quick point about cholesterol, which is another type of lipid later. Let's go for number one, which is the function of lipids. So you need to know the following functions. And actually, I'm going to switch over to the some pictures that I have here to sort of make this point initially, which is the first function of a lipid, which I'm going to put here, is that it can make, or it can be a precursor for various hormones. And the typical example, there are many, estrogen being one, but also cortisol being another. And hopefully you can see here that cortisol, it's sort of very hydrocarbon rich, lots of carbons, lots of hydrogens. And this is one of the major stress hormones in the body involved in the fight or flight response. As you will come across, perhaps in the upper sixth, this is part of the sympathetic nervous system. Which regulates the sort of fight or flight response. The other function of lipids, so a second function here, is involved in the waxy cuticle. So this is a waxy layer on the upside of the leaf. So this is the structure of a typical leaf. And it's a waxy waterproof layer. So what this means is that actually we use lipids a lot of the time, if I move this up here, for waterproofing. This is because they have a structure which we'll come back to many a time, but they are essentially water hating, right? Oil, fat does not dissolve in water. Therefore, it is termed hydrophobic. Phobic meaning to hate, hydro from hydra meaning water. So because of the hydrophobic nature of lipids, they are used to help create a waxy cuticle layer, which you can see here at the top on a leaf, to maintain waterproofing. The third one is shown here, where you have come across, hopefully before, electrical insulation. Electrical insulation. And this comes about because of this guy down here, the myelin sheath. And this, hopefully, you are aware, certainly in your peripheral nervous system, is generated by certain cells. As you can see, they have nuclei here. So the red dots here are actually cell nuclei. Because the cells that wrap their membranes around the axon are known as Schwann cells. At least in the peripheral nervous system, but don't worry too much about that. If you're in the brain, they are made by cells called oligodendrocytes, but we're not going to worry too much about them. But they're called Schwann cells. And the nucleus or the nuclei of the Schwann cell you can see there, essentially, myelin is a fatty substance. It's a lipid that is present within their cell membranes that can wrap around the axon at various points to generate regions of electrical insulation. And hopefully what you remember from your studies across GCSE is that this allows the electrical signal, what we say is that it allows it to jump from node to node. So the regions of the axon that do not contain myelin here, these sort of just purple bits here, are called nodes, specifically nodes of Ranvier. And we essentially watch this action potential, this electrical, from node to node. Of course, it doesn't actually jump in the way that I'm drawing it here, but hopefully that sort of gets the point across. But we're not talking about neurons today. So those are three, just because I have pictures that I think are quite nice. So if I head back to my main notes here, so that was 3. So let's look at 4. So the fourth thing is that we can form part of cell membranes. So both membranes on the surface of a cell, so the plasma membrane or the cell surface membrane, but also membranes surrounding organelles, things like mitochondria, chloroplasts if you're in a plant cell, that kind of thing, the nuclear membrane, etc. And what this allows, if you imagine we have a cell here like this, and perhaps the nucleus is there, this allows for compartmentalization, right? We have a barrier here. So the cell membrane is made up of lipids, and it acts as a barrier. Posh word, compartmentalization. In other words, we can form a sort of inner compartment here in purple and an outer compartment here in red. And that allows us as cells to compartmentalize various metabolic activities which are useful for various different reasons. If you, or as and when you study the immune system and you do phagocytosis, the fact that phagocytic cells can have lysosomes within them containing, various harsh chemicals, hydrolytic enzymes, free radicals, etc. The fact that they are encased here, for example, in a kind of membrane sac like this, means that they are kept from doing any damage to anything else within the cell. So that's kind of what we mean by compartmentalization. The fifth use of lipids is as an energy resource. And that is because lipids, if we do structural formulae like this, have basically lots of carbons and lots of hydrogens attached. Hydrogens not being shown here. So essentially, the reason why it's good for energy is because we have high energy carbon-hydrogen bonds. So these can be broken to, in simple terms, to liberate energy for respiration. That's not quite how it happens. That's a little bit of a misnomer, but it gets the point across. And essentially, you can turn a lipid into an intermediate for respiration. So essentially, we can use it for respiration. Noting here, of course, that we don't just have to use glucose and carbohydrates. We can use lipids as our respiratory substrate. It just has to be converted into another compound that can then be fed into the respiratory metabolic pathways. And that happens because of a process known as beta oxidation. I'm not going to go into any more detail than that. You really don't need to worry too much about it. But if you're interested, feel free to look it up. Essentially, it allows lipids to act as a substrate for respiration. Of course, we can also have thermal insulation for fat. So things, animals use it, for example, seals will use it, blubber, humans use it for thermal insulation. So those are the major ways in which we use lipids. So I'm going to sort of tick that off. We've covered most of those. So I'm going to look now at point number 2, which is a quick recap of GCSE and something called tags. So let's line that off. So this is GCSE, recap, and tags. So at GCSE, we came across lipids before. We came across these in the biological molecules topic, where they were drawn like this. And you may or may not, don't worry if you haven't, you may or may not have been told that this box represents a molecule called glycerol, and that these three chains here, these three hydrocarbon chains, are known as fatty acids. And the reason is they once upon a time were hydrocarbons that contained this functional group, which hopefully we all know as a carboxylic acid. That may be kind of where you got to at GCSE, apart from pointing out, of course, that the point of lipids is that in your diet, you can break these, you can hydrolyze these lipids into the individual monomers. And they get hydrolyzed at this point here. That is quite a big pen. I'll just circle it. They get hydrolyzed here. And what that releases, what that liberates is, of course, your box structure here, which is glycerol. And your three fatty acids. And this was done by an enzyme, which I will typically just draw as a blob here with an active site. Again, recap biological molecules if you're not sure. But this is done by an enzyme called lipase. One of the places, of course, where this acts is in the duodenum, because it is released by the pancreas. But look over digestion and biological molecules if you need a recap of that. So, believe it or not, you've actually covered the next bit that I'm going to tell you, except just not the detail. So this structure here, the three fatty acids and glycerol, we said make a lipid. Specifically, they make a type of lipid called a tri, meaning three, linking to the three fatty acids here, acyl glyceride. So essentially, we've got three acyl groups and a glyceride. That, of course, the three refers to the three fatty acid tails. And the glyceride refers to glycerol. So essentially, all I'm giving this is a name. It's called a triacyl glyceride, or a tag for short. So at GCSE, you'll have covered how that is broken down. It's broken down by lipase, right? Lipids, triacylglycerides are broken down by lipase to produce glycerol and three fatty acids. What I'm now going to do is give you the structure of both the glycerol and the three fatty acids and how this mechanism actually works. So continuing with this theme, let's actually start with the monomers. And I say monomers here, it's very important to note that, of course, lipids are not polymers, but I'm going to refer to the hydrocarbon tails and the glycerol here as monomers, but they don't actually form a polymer, right? Lipids are not polymers because they're not repeating units of monomers. It's not a repeating structure. But the two kind of building blocks, if you like, are our glycerol and fatty acids. So let's look at what these actually look like. Glycerol has this structure. It is made up of 3 carbons, each of which is attached to our hydroxyl group. And then we just simply have hydrogens attached. So that is the structure of glycerol, fairly straightforward. The UPAC name is 1,2,3 propan-triol, or propane 1,2,3 triol. Don't worry about that. In biology, we tend to just call it glycerol. Fatty acids, if I do this in red, all have carboxylic acid groups attached. They then simply have lots of hydrocarbons, or a sort of long hard hydrocarbon tail that goes on for an indefinite, varying lengths. And that, of course, is repeated 3 times. We need 3 fatty acids to join to glycerol. Hopefully, you can appreciate, if I were to draw the same thing here and here, you can appreciate how these are going to join together to form our triacylglycerol. What is going to happen is that the OH groups are going to react in a condensation reaction to liberate water. Okay, that's really important. It's going to liberate water. So let's look at how this is going to work. What you can hopefully appreciate is because we have these OH groups so close to each other, the OH from the glycerol here can combine with the hydrogen from the fatty acids. So I'm going to highlight both of these groups like this. And they are going to be released as a molecule of water. And the water is going to be made up of an oxygen and a hydrogen from glycerol and a hydrogen from the carboxylic acid from the fatty acid tail. So we release water, and that's going to happen 3 times over. We're going to join these together and these together. I will join these just to prove the point that they come off as one. So in total, to join these together, we're going to produce 3 water molecules. Essentially, one here, one here, one here. And the result is that they're going to be joined together. If I now get rid of this, so I'm going to get rid of the OH here. I'm going to get rid of the hydrogen here. And if I fill in the new bond, I'll color it differently to show that it's a new bond that's formed. That is the new bond that is going to be formed. The slight complication here, of course, is that it's going to be bonded to the oxygen on the carboxylic acid tail. So actually, I probably should have drawn it like that. But it's not very pretty, but that's kind of what it looks like. So that's going to happen 3 times. Whoops, gone a bit too far there. Oh well. And there you have your triacylglycerol. So we've got our glycerol here, and we've got our 3 fatty acids now attached. Because we liberate 3 water molecules, this is a condensation reaction. Specifically, it's 3 individual condensation reactions. And it's a specific type because we form this bond. And the bond we formed is an ester linkage. So these in purple here are ester bonds. So we don't just say for lipids that it's a condensation reaction, we actually say it is an example of a specific type of condensation reaction known as esterification. Now, if you're interested in the reaction mechanism for this, I'm not going to go into a huge amount of detail, but I'll very quickly kind of sketch the general gist here. I'll just do it for one of the reactions. But obviously it will happen 3 times over. So this is our glycerol. And it's important to note that on the glycerol, we of course have, It's important to know we have 2 lone pairs on the oxygen here. And of course the carboxylic acid group here. And what's going to happen is the electrons here in blue are going to attack this carbon here. And the reason for that is because this carbon is delta positive because of the electronegativity of oxygen here and the electronegativity of oxygen here. I'm just going to get rid of the delta charges just to make my drawing a little easier. What then is going to happen, not immediately, there is a caveat to this, but essentially what then happens is this bond here then breaks and gives its both electrons back, both oxygen's initial electron and the one from carbon to the oxygen here, and that produces OH minus, and we've formed the bond in blue. So if I draw what we've actually produced, if I draw it in blue here, so lone pair here, the lone pair here has now formed a bond to the carbon, which obviously is connected to the hydrocarbon tail. The electrons in this yellow bond here have been given back to this oxygen to produce OH minus with its electrons now here. And of course, the hydrogen is still knocking around on this oxygen here. What then happens, hopefully you can appreciate, is that the OH minus, it doesn't necessarily happen like this, but it's a nice story, so we'll go with it. If you're interested, you can always contact me for a further explanation. But this lone pair here then attacks this hydrogen because this hydrogen is delta positive, this oxygen is delta negative. This will then pick up this proton, like that. These electrons in this bond get given back to the oxygen to give it back its two lone pairs that it had. And then that produces your ester bond. So the overall product then looks like this, where you've got your oxygen, you've got the red lone pair, it's just been given back. It's got the blue one that it had before. We now have the bond formed to the C double bond O here, which is then linked like that. And that is your ester bond. And of course, your water produced here is a product of that condensation reaction, esterification. So that's a very quick overview. Just to be clear, I have drawn that as a step, showing that step one happens with the first sort of 2 groups at the top there. So this is kind of step 1, this is step 2, this is step 3. But of course, all 3 of these steps happen with each individual molecule here, or 2 molecules, to produce this. So I hope that makes sense. Don't worry too much if it doesn't, because you will never be asked to draw the mechanism. The only thing to point out is, of course, the reverse is true. You can hydrolyze these bonds and you can reinstate the 2 monomers, the glycerol and the 3 fatty acids, by adding water in. So obviously we produce water because it's condensation. But if you were to add water, add 3 waters, then you would hydrolyze these bonds and you would reproduce your glycerol and your fatty acids. And that is achieved, of course, by lipase. Okay, the next thing we wanted to talk about, so let's just tick that off our list. So we've done GCSE rather, recap and tags. We've covered condensation and hydrolysis, specifically esterification. So now I want to talk about saturation, which is point number 4. And the only thing to say here is that the hydrocarbon tails of your fatty acids can be either saturated or they can be unsaturated. And saturated means that each carbon atom is only connected to another carbon atom by single bonds. So we only have carbon-carbon single bonds. And in the exam, please write that as carbon-carbon single bonds. Don't draw it like I have there. You have to actually write the words carbon-carbon single bond. But that's the key point. We only have carbon-carbon single bonds. And I remember this because if you only have carbon-carbon single bonds, each carbon is going to be making 4 bonds, because it has a valency of 4, to other atoms, separate atoms. In the case of unsaturated, we have at least 1 carbon-carbon double bond. And double bonds are shorter than single bonds, and we'll get to that in a minute. So if I were to look at the hydrocarbon tail, so if I were to draw glycerol like that, and I looked at the hydrocarbon tail, you could have a double bond there, for example. You could have another one there, etc. Okay, that would be unsaturated. Let's just draw one in here. So if you have 1 carbon-carbon double bond, we say that you are mono meaning 1, unsaturated. As opposed to something with multiple carbon-carbon double bonds, like this one, which we would say is poly unsaturated. And these are just names you need to know. The importance of the saturation level, right? The number of carbon-carbon double bonds that you have is shown in this picture here. If you were to look at these 2 fatty acids, don't forget you've got your carboxylic acid group here. That doesn't count, by the way, because it's not a carbon-carbon double bond. It's a carboxylic acid group. Palmitic acid, or the conjugate base of palmitic acid, the ionized form, palmitate, if that's how you say that, contains no carbon-carbon double bonds. So this is saturated. Right? Each carbon, let's just pick this one, is making 4 bonds to different individual atoms. If we look over here, you can see we've got a carbon-carbon double bond. These carbons are not making 4 to 4 separate atoms, so it is unsaturated. And because we only have 1 of them, it's called a mono unsaturated fatty acid. The importance of being unsaturated, i.e. having a carbon-carbon double bond, at least 1 of them, is shown here really nicely. Hopefully you can appreciate that this region here causes a bend in the hydrocarbon tail, or a kink, a sort of kink in the chain. Kink in the hydrocarbon chain. It causes a bend in the road, essentially. And this is important when we consider melting point and boiling points. So if I go back to my notes here, you can imagine that I have a hydrocarbon tail like that. If I were to draw, let's say, my triacylglycerol here, triacylglyceride. And let's say that this one is completely saturated. Let's draw another one where we cause some unsaturation. So let's say that's okay. Let's say this one has a carbon-carbon double bond here, and that causes a kink in the chain. Okay, so I'm trying to show that here like that. Okay, it causes a kink in the chain. So this guy down here is mono unsaturated, specifically the third fatty acid tail here. And the importance is as follows. There are intermolecular forces that you've come across called IMFs, GCSE chemistry, for example, that exist between all molecules by virtue of the fact that they have electrons. Okay, so just before we, in fact, actually, I'll explain this first and then I'll talk about why that's true. So these intermolecular forces that I'm going to talk about are called London dispersion forces. Or LDFs. And these are fairly weak intermolecular forces, but they exist between all atoms that have electrons. So I'm going to draw them like this. You get the point. And the key thing is that they can exist between the hydrocarbon tails. So I can draw the same thing here. I'm not going to draw as many just because for time purposes, but like this. The key thing is that when we kink the chain, we can form intermolecular forces up to a point. Soon as you now cause the kink in the chain here, you can no longer have intermolecular forces between these two chains, right? Between this one and this one. The reason is they now exist too far apart, but the bend in the chain has caused the hydrocarbon tails not to stack on top of each other, not to pack as closely together, okay? That means we have fewer, so if I write that down, the carbon-carbon double bond causes a kink in the chain, the hydrocarbon chain. This leads to less close packing. So the hydrocarbon tails pack less closely together. That means we have fewer London dispersion forces. And if we have fewer intermolecular forces, London dispersion forces, that means the relative strength is decreased and less heat energy is required to break apart the London dispersion forces that interact between the hydrocarbon tails. What that means is the more kinks you have, so increased number of carbon-carbon double bonds, this lowers the melting point, which means the more unsaturated you are, the more likely you are to be a liquid at room temperature. So this would be something like butter or margarine, things like that. As opposed to the guy up here that is completely saturated. That means we form the maximum number of London dispersion forces between the hydrocarbon tails. They can pack much more closely together, meaning that more heat energy is required to break all of these intermolecular forces. Don't forget, with intermolecular forces, few of them, so individually rather, they are very weak, but together collectively, they're quite strong. So this kind of a fat here, this kind of a lipid might be something that is sort of fairly solid at room temperature, so something like butter, that kind of thing. Whereas the guy down here is more likely to be a liquid at room temperature. Therefore, it'll be something more like so not butter or margarine, but it'll be something like an oil, olive oil, or that kind of thing. Hopefully that makes sense. I went quite quickly through that because we have actually covered intermolecular forces in terms of hydrogen bonding before. To be clear, this is not hydrogen bonds, because don't forget, a hydrogen bond, something like in water, you need a delta positive hydrogen, which we have here, and you need some electronegative atom here, typically a fluorine, an oxygen, or a nitrogen. Okay, that's what causes a hydrogen bond. We don't have that here, right? There's no delta positive hydrogen near an electronegative oxygen, fluorine, or nitrogen, right? It's just it's just carbons and hydrogens. So although there are hydrogens present, it is not hydrogen bonding. It is London dispersion forces. How they form, I will just briefly explain here, which is, let's just say you have a hydrogen, two diatomic hydrogen atoms here. So I will draw one here and one here. So this is just two hydrogen atoms interacting with each other. What happens is the electrons in the single bond here, we have said previously, exist in the middle of the two atoms. But this is a probability function. It's not that they literally sit there all the time. These electrons, of course, are in orbitals. They're in electron shells and subshells. And it's purely a probability distribution. So, most of the time they spend time between the hydrogen atoms. But in reality, of course, you might get to a stage where the electrons exist very close to this hydrogen. That for an instant in time, right? This is to be clear. This is one moment in time. This makes this hydrogen delta negative, this hydrogen delta positive, as we've seen for other intermolecular forces. But this is temporary. If you were to look a second, millisecond later, the electrons might actually be over the other way, right? The two electrons might be spending a bit of time over here, which would make these poles reverse, right? This guy is now delta minus, this now delta positive. Hopefully you get the point. Let's just imagine that they're very close to this hydrogen here. So we are delta negative here, delta positive here. What that will do is to any molecules of hydrogen gas nearby, that will cause these electrons cause negativity, delta negative on this hydrogen, which then will repel any electrons that are sitting in around this hydrogen. So these electrons here, for example, will get pushed away if these two molecules are close enough, because of the delta negative here causing repulsion. That means these electrons get pinged sort of over here, making this guy delta negative, this guy delta positive, and hopefully you can appreciate all of a sudden we now have delta negative, delta positive, and we can have an intermolecular force. But this is for a brief moment in time. This is temporary. Unlike in water, where we have electronegatives that are constantly hogging electrons, this can happen between any atoms based on the fact that they have electrons, essentially. And the more electrons they have, the stronger the intermolecular force will be. But enough said on that. So this intermolecular force is an induced dipole. We're inducing a dipole, right? A positive and negative end of these molecules. And we call it a London dispersion force. So long as you have electrons, this force can happen. But you obviously have to be close enough to get, right? You have to be pretty close together. Hopefully that's an important point. You have to be close together in space for this to happen. So of course, when you kink the chain, right, you might have a carbon here and a carbon here. Perhaps a hydrogen, hydrogen's attached. They maybe are now not close enough together. I realize I've drawn them quite close together, but that they're not close enough together now for any intermolecular force, any London dispersion force to form. Right, I realize I went fairly quickly through that, but I hope that makes sense. Essentially, how does saturation impact melting point? Well, the more unsaturated you become, the more carbon-carbon double bonds you have. That means that the hydrocarbon chains in your triacylglycerol are kinked. There's a bend in the chain, which means that the hydrocarbon tails cannot pack closely together. This reduces the number of London dispersion forces that can occur between the hydrocarbon tails. And because collectively they're very strong, but individually they're very weak, that means that the melting point will therefore be lower because it takes more heat energy, it takes less heat energy to break them apart. That means that triacylglycerides or lipids that are kind of like this guy down here are more likely to be a liquid at room temperature, something like an oil, whereas some molecule like this would be something like butter, some kind of solid fat, or more likely to be a solid fat at room temperature. Okay, hopefully that makes sense. So that's saturation done. Testing for lipids, very, very quick on this. So this is number 5. So what is the biochemical test for a lipid? This is nothing more than GCSE. You simply do the following. You take, let's say, 2 centimeters cubed of your liquid sample. If it is not a liquid, if it's a solid, something like a bit of cheese or something, you have to essentially grind it up in ethanol to make it a liquid. But assuming it's liquid, so you, by the way, do put that in an exam if it's a solid sample. But if it's a liquid sample, we take 2 centimeters cubed of it. We then need to add typically excess ethanol, however much that is, to the sample. And the reason we add ethanol, you may be wondering, is to dissolve the lipids. Lipids do not dissolve in water because they're hydrophobic, they're fats, but they can dissolve in polar solvents such as ethanol. So don't forget that. What we then do is we add an equal volume of water. So add maybe 5 centimeters cubed or so of water, distilled water. And don't forget, we need to put the positive result. So if it's a positive result, we form a cloudy or a milky or a white. And this is the key word, emulsion. That is absolutely key and something that you have to say. Of course, if it's a negative result, then you wouldn't get the cloudy emulsion happening. The reason this works is because your lipids get dissolved in your ethanol here. When you add water, that essentially dilutes the ethanol. It dilutes the ethanol to the point at which your lipids come out of solution as droplets, and they form an emulsion with the water. So if your test tube looks like that, you will see a sort of cloudy, milky emulsion as a layer, a sort of milky white emulsion forming. If you don't have any lipids in your sample, then of course you won't see this. Okay, that's the biochemical test for lipids done. So what we're now going to do is look at something that is sort of technically, I guess, covered on the other side of the course. But I just want to sort of briefly give an overview of this. I'm not going to labour the point too much because you hopefully have covered this with your other beak on the other side of the course. But it's worth saying anyway. So we're going to now look at phospholipids and membranes, which of course is one of the points, one of the things that lipids do. So this is point number 6, which is we need to know what phospholipids are and their structure. And the key point here is that they are related to the tags, to the triacylglycerides that we've just seen. And they are related as follows. I'm actually going to draw the whole thing out, so I apologize for the delay here. Feel free to speed the video up. But I am actually going to draw right. So this is our triacylglyceride, where of course this whole sort of section here is made up of glycerol. This is sort of the fatty acids making our typical tag that we saw earlier. One point I just want to make out, I've deliberately put a carbon-carbon double bond in here to show you that these three fatty acid chains do not have to be the same. They can be different fatty acids. So some of them can be saturated, some of them can be unsaturated, even within the same triacylglyceride. Key point is though, we can modulate this now. We can change this to form something called a phospholipid. And as the name suggests, all we do is add a phosphate group. And we can add a phosphate group by doing the following. The 2 electrons that are involved in this covalent bond, you don't need to know how, but we essentially break this bond and give them both back to this oxygen. Okay, so we hydrolyze that bond. Don't forget, good terminology, hydrolysis, using water, right? We would need water in order to do this. There'd be an enzyme that does it. And we can give back those electrons to this oxygen. So it has its both lone pairs now, right? It had a lone pair anyway, like that. I'm just going to get rid of those for now. It doesn't really matter too much. I'm going to get rid of this because we've now broken the bond. And the 2 electrons that we're going to focus on that we've just regained are these 2 here. And the key thing is these can now be used as a nucleophile to form a dative bond with something else. And in this case, it's a phosphate group. Okay, so a phosphate group, hopefully we appreciate, looks like this. This is phosphoric acid, and it becomes phosphate when it gives up its 3 protons, 1, 2, 3, like that, giving each of these oxygens a negative charge, and it forms PO4 3-. It's an ion. So we can now draw this attached to our triacylglycerol because we have triacylglyceride in this case, because we've now broken the bond, given the electrons back to oxygen, and we can now link this to a phosphate group. Like that. Now, if we were drawing this as our box and sort of general line structure, I would draw the glycerol like this. I would draw my 2 fatty acid tails like this. And the phosphate, just to make the point, we actually sort of orientate it differently on our molecule. We kind of stick it at the top here in a circle and we call it PI. PI standing for inorganic phosphate, right? That's this group here. So this is now a molecule where we have a phosphate group. We have our glycerol. And we have 2 fatty acids, 2 fatty acid tails. All of this is now called a phospholipid because it's a lipid, as we've seen, but now with a phosphate group attached. And actually what we tend to do is get rid of the glycerol in our notation. We tend to be a bit lazy and just draw the phosphate head and 2 fatty acid tails. We kind of ignore the glycerol, but it is there. And this is the structure of a phospholipid, okay? And because, of course, this PI group is, if I draw it like this, don't forget it's negatively charged here. It's connecting to oxygen, which is connecting to the glycerol. We have negative charges. So the phosphate head here is charged. And what that means, hopefully from the biochemistry stuff we did before, this means it is polar. And if you are polar, you dissolve in polar things, such as water. So this makes the head, the phosphate head, hydrophilic. Okay, water, hydrophilic, phile, meaning loving. So it dissolves in water. It's hydrophilic. It's a hydrophilic phosphate head. The hydrocarbon tails, of course, are hydrocarbons. They are fatty acids. Fatty, therefore they are hydrophobic. No change there. So we've now created a molecule that is both hydrophilic, it both kind of loves to interact with water, and it hates to interact with water at the same time. It is therefore confused. Right? It doesn't know, it kind of can interact with both, right? It doesn't know is it hydrophobic, is it hydrophilic? It's a bit of both. And the posh word for being both hydrophilic and hydrophobic isn't confused. It's amphipathic. Amphipathic, posh word, meaning you are both in the same molecule. The same molecule is both hydrophilic, water loving, and hydrophobic, water hating. Now, what that means for us is that if you were to put your phospholipids in water, they would arrange themselves spontaneously like this. Where we have water on the outside, but water is excluded from the center. So, remember, which bits of the phospholipids here are hydrophilic? The answer is it's the phosphate heads. It is these guys that are going to be interacting with water. Same down here. In the middle here, the tails, these are hydrocarbon tails, they're fatty acid tails. We are therefore very hydrophobic. So no water, right? Hydrophobic in here. So this whole bit is hydrophobic. Of course, the phosphate heads are hydrophilic. And you can imagine, therefore, that this is actually part of a larger structure. So if I get rid of this, that these lipids actually continue, right, to form this structure. Apologies for the bad diagram, but something like that. You imagine they keep going in this direction, keep going in this direction. Your cytoplasm, you've got water on the outside, which is kind of in the extracellular space. And you've created two compartments, an inside and an outside, right? So this could be the cell membrane. And because it forms this structure, we have two leaflets, right? We've got this leaflet, this kind of half, and we've got this half. It is therefore termed, the cell membrane, or certainly any sort of membrane where you put these things in water, it is called a bilayer, two layers, right? We've got an outer bit that interacts with water, and the sort of inner bit also interacts with water, the phosphate heads, but you've got two layers that are doing so. And that is the structure of the cell membrane. So cell membranes, or actually any type of membrane, so organelle membranes as well, form a bilayer structure spontaneously because the hydrophobic tails here in the middle in red will pack together to try and get away from water as best they can, and the hydrophilic heads will interact with water. So that is the structure of a phospholipid, and the fact, or I'm giving you the fact that they spontaneously aggregate to form bilayers, and this forms cell membranes, and indeed any membrane of organelles, which allows for compartmentalization, and essentially forming the inside of a cell and an outside, or the inside of a organelle and the cytoplasm on the outside. And this structure is called a phospholipid bilayer. The phosphate heads are hydrophilic, and the center of the majority of the bilayer is hydrophobic. And that's an important point that I'm going to come on to next. So let's just look at the menu and tick off. So we've done phospholipid structure, we've covered membrane structure. I now want to cover transport. That's point number 7. So point number 7 is transport across membranes. As we can see here, if I kind of try and tidy this up, the membrane itself, the kind of inside, the sort of inner bit here that I'm highlighting in purple, this bit, is completely hydrophobic. There's no water here at all. Water cannot interact with this. Anything that's hydrophilic indeed cannot. So the key thing that I want you to remember here is this whole kind of like dissolves like phrase, which means that essentially if you're going to dissolve in water, you have to be hydrophilic. If you want to dissolve in something, so the reason why that's true, of course, is that water itself is polar, and polar things therefore can dissolve other polar things. If you're hydrophobic, that means you can interact with other hydrophobic things, but you cannot interact with hydrophilic things. So let's draw the membrane. In fact, I'll just show you the picture. It's probably easier. This is a little bit of stuff about phospholipids. I'm not going to really cover it too much. I may as well just point it out because I put it here. Not only can you have your phosphate attached to your glycerol, you can actually attach another group on top, which here is shown as something called choline. Okay, it's the same thing, but just shown as the real chemical formula here. Nothing to sort of shout home about. You don't really need to worry too much about it. But essentially, you can change this choline group for different things, and you can therefore change the flavor, if you like, of phospholipid that you can have in a membrane. Nice picture as well, because it shows the double bond causing a kink in the chain. That means that our van der Waals forces can exist here, here, cannot exist any further. The reason I'm showing you this is because there are two flavors that I tend to talk about in lessons, which you'll have heard me blabber on about. Phosphatidylcholine, which we've actually shown here, because this is a choline group attached. Therefore, that makes this whole phospholipid a phosphatidylcholine. You can have another group called ethanolamine, makes phosphatidylethanolamine, and that's a much smaller group. And what it shows here is that means that makes this phosphate head here a lot smaller, and that means that they can pack just that little bit closer together on that side, and that allows the membrane to actually curve and kind of bend around. It doesn't just remain as like a rigid flat structure. So if you're ever wondering how the bilayer actually bends around to form the cell, which I've just drawn in the previous section, that's how it does it. It varies the composition of the different types of phospholipid within the membrane by varying the group that it sticks on here and here to the phosphate head. I didn't talk about that. Your exam board doesn't talk about it. You don't need to know it. For our purposes, we just focus on the phosphate and kind of leave it at that. So don't worry too much about that. I'm going to show you this instead of drawing it all out. And the point here is this. This grey bit here is the membrane. It could be the cell surface membrane, or it could be that of an organelle. And it is hydrophobic, right? And we now know that it's hydrophobic because, of course, the phosphate heads are sticking out here. And it's the tails, the hydrocarbon tails that are packing in here tightly together. Oops. Like that. And heads on the other side, right? This is the bilayer. So all of this stuff in here is very hydrophobic. So the whole membrane we refer to as hydrophobic. It's lipids, right? It's a fatty layer, essentially. This leads to something called selective permeability. Now, you should have covered this in the other side of the course, so I'm going to sort of skip through it relatively quickly. I'm not going to labour any points or anything. If you have questions, feel free to email me or message me. But essentially, this limits what can pass through the membrane. That is because it is hydrophobic. That means the only things that can pass through are either gases or other hydrophobic things. Remember, like dissolves like, or, you know, like interacts with like, right? So polar things like water can dissolve other polar things, right? Like ions, things with OH groups, delta charges or formal charges. Something that's hydrophobic or fatty like this can therefore only dissolve other hydrophobic things, right? That don't have delta negatives or delta positives or formal charges, right? So hydrophobic molecules, right? What we mean by this is no either partial, and of course by that I mean delta, or formal charges. Okay. So glucose obviously is not one of these, right? Because you've got OH groups knocking around. And the key thing is that gases, so oxygen, carbon dioxide, nitrogen here, for example, absolutely can pass through no problem, right? So they are not limited in any way by diffusion. They can just, this is diffusion, by the way. That's how we're getting across here. Diffusion allows them just to move across, okay? Because they're small enough, not sort of polar enough, I guess, but certainly they're small enough to pass through. Steroid hormones, so things like, we talked about cortisol, things like estrogen. These are your sort of steroid hormones. These are hydrophobic. That means they can also pass through just by diffusion, right? Passive diffusion, no problem. They are not hindered in any way getting across the membrane. That's because they can interact with the fatty acid tails, because the interior of the membrane is very hydrophobic. Now we get to sort of more complicated areas. So let's look at the next one first, right? It doesn't matter about size here, really. There is a caveat to that. Eventually, it kind of does, but it doesn't really matter about size here. If you're hydrophobic and you're small enough, you're absolutely fine. You can get through. We then have, instead of being hydrophobic, we can have polarity. Okay, polar molecules. So I'm going to highlight polar molecules here and polar molecules here. Okay, what this means is we have delta. Actually, I'll just draw it out of this. What we mean by these is we have delta charges. Okay, not formal charges like ions, but we have delta charges. So glucose, obviously, down here is one of those guys. But obviously water as well, of course, you know, delta negative oxygens, delta positive hydrogens, glycerol, for example, we've come across, ammonia. We have delta charges. And essentially, these are limited. Eventually, you'll get some diffusion happening. Okay, it's not that they are completely excluded from the membrane, although we kind of tend to just assume that they kind of are. But actually, water can get through to a limited extent, as can urea, glycerol, and ammonia. But the key thing is the majority of them, the majority cannot, right? So this arrow here is showing that the majority of the time they can't get through. Occasionally you get a little trickle of them moving through. Okay, so you might get a little bit of water moving through. You might get a little bit of urea, etc. Okay, but that really isn't very common at all. And of course, the bigger you get, the more of a problem that is as well. Okay, so glycerol, far less of that will trickle through than something like urea or water. Hopefully that kind of makes sense. That is because these are slightly polar, which means that they are not hydrophobic, right? They're actually hydrophilic to different degrees, right? And it depends how much electronegativity and positivity you have, how much polarity you have, as to how quickly and efficiently you can get across the membrane. But it's very limited. We can then move down here and we can go, okay, what about if you're large and polar, right? And that's even worse because we're now getting to a position where not only are you polar, so you cannot interact with the phospholipid tails, which kind of excludes you anyway, but you're also basically too fat to get through anyway. So even less, which is shown by this really thin arrow, even less of, molecules can get through. Occasionally you get some, maybe, but very, very much rarer that happens. The majority of the time, these are completely excluded and cannot get through. So examples are sugars, right? So glucose, for example, cannot pass through the membrane to get into or out of cells. The absolute rule on this is that if you're an ion, you are not just polar, you are charged. Right? And if you are formally charged, you have positive or negative charges, there is absolutely no way you are getting across the membrane at all. Okay, so these are kind of the extreme of being polar. You're not just polar, you're charged. Okay, so you can see here that there is absolutely there's not even like a background trickle of stuff that gets through, right? They are completely excluded. So hopefully that kind of explains what we mean by, at GCSE, we used to call it this. We didn't really explain it, but selective permeability, right? The membranes, cell membranes, organelle membranes are selectively or partially permeable. And that's what we mean, right? We're essentially selecting against all of these things here. And that begs the question, though. Normal diffusion can happen here, so that's no problem. How do we get stuff across the membrane if you're in either of these three boxes, especially ions as well? But, something like glucose, we actually want glucose to get into the cell, right? Glucose is used for respiration. So if you can, if only a very, very slight trickle gets through once in a blue moon, then that's not going to be enough to allow the cell to remain metabolically active and, produce enough ATP for its functions. So all of this is a problem, right? Water as well, right? You actually need a decent amount of water being able to be moved in and out, and especially ions, right? Think about nerve cells, for example, right? How do you get sodium ions across? That's the whole reason that axons and neurons can act as transducers and essentially conduct electricity is because they can conduct charge, right? The movement of ions causes charge, and they can't do that if sodium ions are blocked from moving across the membrane. They need sodium ions to cause that electrical current. So the answer to this is as follows. If you can diffuse across the membrane without help, right? We need no help here, no help needed, right? This is normal diffusion, and we call it just simple passive diffusion. Okay, that's kind of what we've talked about before at GCSE. It's just diffusion, right? From a region of high movement of net movement of particles from a region of high concentration to a region of lower concentration down a concentration gradient. Okay, so of course, if you start to build up concentrations of oxygen over here, you'll start to move back, until a sort of dynamic equilibrium is reached. But anyway, so how does this happen? Well, the answer is you still rely on diffusion. This is still diffusion, right? This is diffusion, this is diffusion, this is diffusion. We rely on concentration gradients, right? A high concentration over here, lower concentration here, we will move in that direction. If we reverse that, then they'll move in the other direction. All well and good, except they can't actually get across the membrane in the first. It doesn't matter about concentration gradients at the moment because you can't get across, right? So the answer is diffusion here can only be allowed to happen if we do something to facilitate movement across the membrane. So this type of movement is called facilitated diffusion. Right, and the key point is it's still diffusion, right? No energy required. That's really important, right? Still diffusion, right? Except it's not really sort of a passive process anymore, really. I mean, it kind of is, but we refer to it as facilitated because we need to do something. We need to make the membrane permeable or more permeable to these molecules. Okay, we need to do something to allow these, let's say, ions, for example, down here through. And the sort of short, sort of quick answer is we do the simple thing of essentially puncturing a hole in the membrane somehow, and we create a region here, either side of this channel, a region of hydrophilicity, a region of hydrophilic philicity. Okay. And what that means is that then the ions, the charged things, can then move through here. We're creating a region of space in the membrane that they can move through, and that allows diffusion to happen, right? So long as, providing we have a high concentration of something like sodium ions here, and we have a lower concentration of sodium ions here, we will move from left to right. Of course, if we reverse those concentration, that concentration gradient, we'll move from right to left. Hopefully that makes sense, right? All we're going to do is we're going to allow diffusion to happen, right? It's not active transport. We're not going to do anything weird. But we do need to kind of alter the membrane chemistry somehow to basically punch a hole in it and make this kind of hydrophilic pore that allows the molecules to go through. And that's exactly the same thing that we do for glucose and sucrose and water and that kind of thing. That's the sort of in a nutshell bit. And what I'm going to show you now is the two ways in which we do it. So I should have some other images somewhere. Here we go. These are the two ways in which we do it. We use proteins. Okay. So these green things here in our membrane are proteins. Okay. And it's the proteins that provide a area of membrane that is hydrophilic to allow hydrophilic things to pass through. The easiest way is to imagine it like this, right? So this is our protein. It's essentially puncturing a hole in the membrane and it acts as a channel. So we call this a channel protein. Okay, it inserts itself into the membrane, right? If you were doing a bird's eye view of the membrane, it might look like that. It kind of inserts itself as like a hole here that our ions now can move through like that. And that is because it creates a region of hydrophilicity here. Okay, so this kind of inside the channel is kind of quite nice for charged things to move through. So this is a region of hydrophilicity. So it's a hydrophilic interior. Okay. How they do that, hopefully you'll appreciate when we do protein structure. It links to the R groups of amino acids that are used in the protein that themselves are hydrophilic. But anyway, don't worry too much about that for now. The key thing is that it's a channel protein, right? It's essentially a hole in the membrane. Okay. Now, that hole can be closed or open by I don't want to go into too much detail here. Essentially, you can put like a gate on here, right? A sort of metaphorical gate, and you can either have it closed like this, or you could have it open like that. Okay, so just be aware, it's not just a hole in the membrane that kind of sits there as a hole. Some of them are, but you can either be always open, or you can be what we call gated. Okay. And I want to just give you the information for next year. In upper sixth, when you study neurons and action potentials, an example of channels that are always open are potassium leak channels. And examples of gated channels are things like your sodium and potassium channels that are voltage gated. So voltage gated sodium ion channels are a good example of something that's gated. In other words, that they only open under certain circumstances. But essentially, it's a hole in the membrane, right? You either just cover the hole up and open it, remove the covering if you want to allow it to be gated, or just leave it always open in the case of a potassium leak channel. But that's next year, but I just wanted to point it out. So just to be clear on this, all of this region here is hydrophobic. As we know, but this protein here now causes a nice bit of stuff or a sort of channel within the membrane here that's hydrophilic. That allows passage of things like ions, glucose, water, blah, 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 through. That is a channel, oops, absolutely the wrong pen. Come on. OneNote's being weird. Anyway, point is that is a channel protein. The alternative, by the way, just to point out, these are still specific. Okay, these are specific for certain molecules, right? It's not a hole that everything can pass through, right? Even if it's open, it is specific for water or potassium ions or sodium ions. Okay, how that is true, you do not need to know, and I'm not going to spend time covering it now. If you're interested, message me. It is interesting actually how it does it, but anyway. The other option is where you have something like this, which is not a channel, but it's called a carrier protein. And essentially, it inserts itself in the membrane, but it's exposed on one side of the membrane, right? So you can see that this bit is kind of exposed on the solute binding side of the membrane. So here you have your solute, whatever that is. So this could be a sodium ion, for example, or it could be glucose. Actually, let's use glucose as an example. And glucose binds to the protein, right? The binding site to glucose is on one side of the membrane. When glucose binds, it triggers a change of shape in the protein carrier. Okay, we don't need energy for this, but binding alters the chemistry of the different amino acids within the structure, and it causes a change in shape. And that closes the side of the protein that interacts with the bottom half of let's say it's the inside of the cell or the outside of the cell even, and it opens, as you can see here, it opens a region that's on the other side of the membrane, and that allows the solute to then diffuse out down its concentration gradient that way. The change of shape going from here to here, we give it a word in biology, or a couple of words, really important. I'll write it here. It is called a conformational change. Conformational change. So binding of your solute, whether that's glucose or fructose or whatever it is, ions even, that binds to the carrier protein, causes a conformational change within its 3D shape, linking to the next topic, its tertiary structure. And it sort of closes the binding site on one side of the membrane, and it opens up another kind of binding site, I guess, on the other side of the membrane, or it exposes the solute to the other side of the membrane. That's what I'm trying to say. So hopefully you can see that it's come in this way from this side, and now it can exit this side. That is the difference between a carrier protein. These are carrier proteins that do this, right? They have conformational changes that allow the molecules through, as opposed to just being a hole in the membrane that you can either gate or not. It is really important to point out that both of these ways, we don't need energy, but we do need a facilitator, right? The facilitator here being some kind of protein, either a carrier or a channel, that is embedded within the membrane to create a region of hydrophilicness that allows passage of whatever specific solute it's specific for. This is therefore facilitated diffusion, right? We still rely on diffusion, right? If you were to increase the concentration, let's say here, if you were to increase the concentration here on this side, your stuff would start to move back through this way. Oops, that's not working, is it? There we go. We're still, using diffusion, right, from a high concentration gradient to a low concentration gradient. That is different to something called active transport, which goes against a concentration gradient. So just to show you that, I'm not going to talk much about that at all. This is kind of what we've done, right? Simple diffusion here. This is things like gases, hydrophobic stuff, right? We've covered that. Concentration gradient here from high to low. We can then either be channel mediated or transporter mediated. So this is facilitated diffusion rather than just simple diffusion. And all of these are passive. And by passive, we mean no energy is required. That is in contrast to active transport, which is this one. So this is called active transport, because we're going to force this molecule here. Hopefully you can appreciate we've got a low concentration here, much higher concentration here. We're forcing it against its concentration gradient. So we require an input of energy, right? Typically ATP. Okay, we need to use ATP to essentially pump or force this molecule against its concentration gradient, because diffusion, of course, would want it to go the other way. Enough said on that. I'm not going to labour the point. It should be covered by on the other side of the course. Eventually, I'll get around to doing videos on the other side of the course, but for now, I'm sticking to the beak one side. So this is the biochemistry stuff, really, rather than just the transport. But I've already talked for long enough about this, but hopefully it gets the point across. So was that everything I wanted to talk about? I think it was. I now just want to mention, though, because we've talked about proteins in the membrane, I want to talk about this membrane now. So this is the lipid bilayer, it's the phospholipid bilayer, as you can see, right? Phosphate heads in blue, hydrocarbon fatty acid tails in yellow. The sort of inner and outer bit of that membrane. So it's the lipid bilayer, phospholipid bilayer. But what this diagram shows nicely is that we've got loads of proteins in the membrane. And you can have proteins that are attached on the periphery, meaning the outside of the membrane, right? They might be anchored to a phospholipid somehow, right? As you can, or in this case, sort of hydrocarbon tails. This one here is covalently linked to a lipid, right? Whatever the lipid that may be. But they're peripheral proteins. They sit on one side of the membrane and are linked into kind of one leaflet. This is different to something like an integral protein here, which is where we have a region of the protein that spans the entire membrane and it connects to both sides, right? You've got a bit of the protein sticking out this side, a bit of the protein interacting with this side. So that might be something like the channel or the carrier protein that we've seen previously. They're called integral. The only other, you don't need to know anything else about them, right? Just that's what they're called, fairly obvious, right? You're either on one side of the membrane sticking out, or you're interacting with either both leaflets or one leaflet of the membrane, in which case you're integral. This is just to point out, though, aside from phospholipids in the membrane, we have sterols. So we'll come to what they are in a minute, but you have other types of lipids, not just phospholipids. You can have phospholipids here that are connected to sugars, which, have I got an example of that? Yeah. So you can see that here, right? This phospholipid on the phosphate head is connected to carbohydrates, those hexagons here. And you can see another one here, and it's labeled as a glycolipid, right? Glyco meaning sugar, lipid meaning lipid. So if we're attaching carbohydrates to our phosphate heads, it's called a glycolipid. In a similar way, we can have a glycoprotein where we stick carbohydrate groups here onto our protein. So you can have all of these different types of things in your membrane. It's not just lipids, as far as the eye can see. Not that your eye can see it, but anyway. It's not just a case that your lipid bilayer is really boring. It's actually full of stuff. And the only other point to mention is because of the chemistry, which we'll talk about in a minute, of the phospholipids, it's actually moving around quite a lot. Okay, it's quite a fluid structure. Okay, it's not rigid at all. And this allows, obviously, for the cell to kind of move around, bend, form membrane sort of folds and that kind of thing. So we call this entire model, right, the fact that there's loads of stuff in it, right? It's like a mosaic. Lots of proteins, lots of glycolipids. It's not just boring phospholipids that sit there, and it's fluid, right? It's moving around. So we call it the fluid mosaic model. Okay, that's why it is described as a fluid mosaic model. Right? It's just a word we've come up with as biologists to explain, the structure of a membrane. But it's referred to as the fluid mosaic model. The only other thing just to mention is examples of our peripheral proteins and our integral proteins and the other words for them. So just to be clear, periphery obviously sits on the periphery of the membrane. It's kind of either anchored in there by something or it interacts non-covalently, for example, with the phosphate heads. Integral proteins span at least one membrane leaflet, very often two. And I want to give you examples of these. So the first thing is that they can be called integral proteins, can also be called intrinsic. And peripheral proteins can also be called extrinsic. So just be aware these are two synonymous terms for that. Obviously, the intrinsic ones we've said transport or carrier proteins. So carriers or channels are good examples. Extrinsics tend to be, these can be enzymes, perhaps receptors in some cases as well. So nothing more really to say on that. The final thing of this lesson is to talk about this guy, sterols, which are quite common in the membrane, and they're there for a reason. The example you need to know of a sterol is cholesterol. Okay, and aside from doing this in membranes, it is also a precursor to vitamin D and bile salts. Okay, so if you ever asked, what else can cholesterol do or make, that the answer is vitamin D and bile salts. But anyway, looks like this, and hopefully you can appreciate, right? We've got lots of hydrocarbon rings here that make the structure quite planar. By planar, I mean kind of flat, right? Almost two-dimensional. And the thing is that cholesterol does is not only is it a precursor to bile salts and vitamin D, it regulates the fluidity. Right? It regulates membrane fluidity. And of course, we've talked about the fluidity with regards to the fluid mosaic model. Now, why is the membrane fluid, first of all? There are two reasons why it's fluid. It's kept fluid by cholesterol. But the other reason is the saturation level. Because don't forget, if we have our two phospholipid tails like this between our phospholipids, and one of them One of them is kinked, right, like that. I'm kind of over, let's imagine they're both kinked, right? I'm over exaggerating this. What that means is we have less Van der Waals, that's the sort of poor term for it, London dispersion forces between these hydrocarbon tails, right? They don't exist here. What this means, of course, is that we are weaker because, you know, if you have fewer Van der Waals forces, that means there's less energy to break them apart, which means that these two phospholipids here, hopefully it doesn't take much imagination to kind of see that they're going to kind of slide relative to each other, because these Van der Waals forces here, because there aren't many of them, can break a lot easier. So you'll be constantly breaking and reforming them, right? So these phospholipids are going to be moving pretty, fluidly relative to each other. Cholesterol helps with this because there's a problem here. You don't want to be too fluid. So the problem is at high temperatures, first of all. How does cholesterol maintain fluidity? Right, cholesterol, that's the whole role of this. It helps with fluidity. It maintains regulation of fluidity. Right? Now, at a high temperature, you can imagine we have a lot of kinetic energy. That means that we're going to be breaking and reforming London dispersion forces quite a lot, and the phospholipids and the tails are going to be moving a lot more and a lot faster. And you're in danger of essentially having enough kinetic energy to break these London dispersion forces and kind of disintegrate the membrane. Essentially break the membrane apart, right? So that the phospholipids are kind of no longer interacting with each other, and we now have a hole in the membrane. Cholesterol stops this because cholesterol kind of wedges itself in between the phospholipid tails, and it acts as an anchor upon which, or to which, we can form London dispersion forces. So if we have a look here, it kind of shows it here, right? So cholesterol can wedge itself in between. So just because these phospholipids are moving quite fast relative to each other, you actually put something in between them that stops them from kind of moving too far apart from each other. And the reason that it can do that is because it provides a region that's close enough to each of these phospholipid tails to form London dispersion forces too, like that. Otherwise, you know, most of these forces, these London dispersion forces, wouldn't be able to form, and therefore these two phospholipids here would separate from each other. Okay, so that is how cholesterol at high temperatures maintains kind of rigidity. It kind of regulates fluidity. Okay. So the problem is we're too fluid at high temperatures, just to be clear. The problem is we're too fluid, potentially, if the temperature's high enough. That is bad because we will break up the membrane. So cholesterol wedges itself between the phospholipid tails. It acts as an anchor for more London dispersion forces to essentially keep the membrane from being too fluid. Keep it kind of somehow a bit more rigid than it would be without. I hope that makes sense, right? It stops the membrane from becoming too fluid at high temperatures. What about at low temperatures? And this is the final thing for the lesson today. So what happens at low temperatures? Again, we have reduced kinetic energy. So that means that the phospholipid tails here can pack very closely together, and we form all maximum number of London dispersion forces here between them, and therefore they're very strong. But the problem is if you have too many London dispersion forces here, that means that the too much energy is required to break them, and they don't actually break enough to maintain any kind of fluidity. Okay, it would be too rigid. Okay, so at low temperatures, we would form the maximum number of London dispersion forces between the tails, and that means that too much energy is required to break them apart to get any kind of fluidity back, right? It would be a very rigid structure, right? So what cholesterol does is cholesterol, again, wedges itself between the phospholipids. So cholesterol in yellow here in the middle. And what that does is it physically separates the phospholipid tails, right? It stops the phospholipid tails, hydrocarbon tails on the phospholipid, from packing too closely together. And that limits the number of London dispersion forces between them and therefore maintains fluidity in the membrane. So, oops, going a bit weird. Right, if I go back to my lipids here, we've covered a lot there, probably a solid week and a half, 2 weeks of teaching, but that's now covered transport of the membranes and cholesterol.